Hello everyone. It's a pleasure to have you online for this third webinar in UNIgreen's Biochemistry and Biotechnology series. And today we will have a talk about within the topic of biotechnology applied to agriculture. Before giving the stage to our speaker today, just a few forwards. I will be your host. So I'm Patrick Gonzalez. I'm Associate Professor at Biotech, and I'm co-coordinator of research and innovation for the UNIgreen University, who is organizing this webinar. You have here my email in case you would like to contact me for any question. And as a disclaimer, I would like to mention that this webinar is recorded and it will be uploaded in a UNIgreen YouTube channel as our previous webinars. And if you are a student and you would like to get a certificate for attending this webinar, just contact us at this email address you can see here. Please feel free to ask any question in the chat, and after the talk, I will try to unmute the microphones and we can speak directly. About UNIgreen, because you might not be familiar with UNIgreen. So UNIgreen is an alliance, European alliance of 8 higher education institutes across Europe, supported by the Erasmus Plus program. And it has the particularity to be dedicated to education and research in the field of sustainable agriculture, green biotechnology, and environmental and life science. And our goal is to deliver, of course, education, but also education through research and provide opportunities for both student and researcher to make exciting research projects. So here are all the partners within the UNIgreen, and you can find here our website address to get more information about our activities. Within research, we have 8 different topics. Today, we are, for this webinar, in the third topic concerning biochemistry and biotechnology, but we have 7 other topics that also will deliver some webinars. So if you are interested by these topics, just stay in touch, go on our website to get new events and news about these webinars. Just as a reminder, 2 webinars were already done in this JRC, Biochemistry and Biotechnology, and you can find them on our YouTube channel if you want to see these webinars. So today we have our third webinar, and to come in January, February, and March, you can see 3 other webinars with the different topics that will be explored. So coming to today's agenda, so we will have one presentation in the topic of microbiological biotechnology applied to agriculture, and then we can have some discussion, conclusion, and closing. About our speaker today, so we have really the pleasure to welcome Professor Stefan Shilev. Stefan is Director of Research at the Plovdiv Agricultural University, and he is a specialist in environmental and agricultural microbiology. What's also interesting is that he is also interested by circular agriculture and bioeconomy, and he also developed interaction with public stakeholders, in particular municipalities, for example, which is very interesting because it's allowing to push research to real situations, so on the field. So with this, I would like to thank again Stefan for joining us today, and I will let you the stage, Stefan, for your presentation.
Thank you, Patrick. First of all, I would like to thank to Biotech for the invitation to join the webinar, the third webinar. So now I would like to give you some insights on the role of microbiome, especially soil microbiome in circular agriculture. So my name is Stefan Shilev. I'm professor at the Agricultural University of Plovdiv on microbiology and environmental biotechnologies. So today I will present you some introductory information, and after that part of our work on one of our projects. First of all, you know, maybe you're familiar about the intensive agriculture and the pressure of intensive agriculture on the environment. Although we have considerably improved productivity from the conventional to integrated agriculture and have new crop varieties available, we still depend too much on monoculture, mineral fertilizers, and synthetic pesticides, setting different problems as stressing the environment, leading to different problems with energy and matic fluxes. At the same time, we neglect plant microbiome interactions, while the diversity is suffer different problems and decrease. The prolonged and substantial use of agrochemicals such as mineral fertilizers, pesticides, conducts to different consequences, such as environmental impacts. We can say that 1/3 of the greenhouse gas emissions from agriculture, they damaged as climate, air, soil. They conduct to water pollution. The agriculture extensive agriculture conduct to a significant loss of biodiversity. Different health concerns, soil depletion and degradation, because a lot of nutrients are lost, such as agriculture, such as organic matter. They are lost in the underground. From that side of view, we have decreased soil biodiversity, but also different problems with animal welfare. Of course, also pesticide resistance, antimicrobial resistance. All this is due to excessive use of chemicals in agriculture. So you may observe here that in this picture you can observe almost most of the negative effects of intensive agriculture, as nutrient runoff, leaching in the underground, air contamination via ammonia emissions, and climate change problems with mostly nitrogen, nitric oxides, and also CO2. During the last decades, arose circular agriculture as a way to overcome the problems in agriculture and, on the other hand, in environments. The circular agriculture is not quite new. It's a not new idea, new approach. Its goal is to promote high quality food, increasing the sustainability through environmental friendly approaches. It is focused on valorizing agricultural byproducts and their use in agriculture. Again, this is the principle of 3R, reduce, reuse, and recycle. Integrating crop and livestock, because a lot of farms, there are a lot of farmers, they are specialized in one or in the other kind of production, but combining integrating crops and livestock production is crucial for circular agriculture. So in circular agriculture, we have also another benefits such as increasing soil biodiversity, but we need to apply bio-based solutions to reduce the use of chemicals. So we know that we cannot avoid the use of agrochemicals, but at least we are obligated to reduce these inputs through using biofertilizers, biopesticides, combining different crops, using also crop matching. I mean, matching one or the other crop, using growing these crops in the same place in the same time. That is known as intercropping, and etc. approaches. So in this picture, you can observe the idea of circular agriculture. It could be applied also in different stages of our life, but generally it's a part of circular bioeconomy, using processes like recycling, bio-based approaches, etc. Concerning the role of microorganisms in agriculture, we can say that this is one of the main approaches of circular agriculture. Use bio-based products to substitute or minimize the chemical inputs. In this respect, the introduction of organic amendments is a valuable approach to obtain these goals. Why? Because applying organic matter, recycled organic matter, on one hand, we have avoided the accumulation of waste, while from the other hand we supplied different organic sources for plants, and especially for the microorganisms. The soil microbiome, especially in the rhizosphere or in the same plant, is dependent on these nutrients that we supply. So the incorporation of agricultural or animal waste or compost or other biofertilizers could modify the nutrients by could support the role of microbiomes to modify these nutrients and to convert them to mineral nutrients that are mainly used from the plants. The beneficial microorganisms are also presented in the soil, so they could modify the states of the nutrients and support the plant growth and development. On this figure, we can observe approaches to convert the plant residue to nutrients for the plant and for the soil microorganisms. On the first place, this is the composting and vermicomposting approach. That is considered one of the most suitable recycling methods to valorize waste in agriculture. Another one is anaerobic digestion. Other with also some importance, but not so considerable value from the point of view of environment. This is the pyrolysis or incineration producing ash or biochar. From this starting point, I would like to say some words about our work done in ConnectFarms project. So the name of project is Circularity Mixed Crop and Livestock Farming System with emphasis on climate change mitigation and adaptation. It was financed by Horizon Aeronet program, and it is also in line with different sustainable development goals of United Nations. We are 10 partners. Actually, the project finishes some months ago from these countries among universities and research institutions. First of all, our work started with valorizing agricultural waste through composting, followed by vermicomposting. So the compost was composting pile was produced from cow manure from of 4 tons, wheat straw 1.5 tons to consider to obtain carbon to nitrogen ratio of 26. So the humidity was maintained during the process at 50-60%, with turning the composting pile to introduce oxygen and to decrease the temperature. The duration was 4 months. After that, the compost, which was not really mature in that stage, was used for vermicomposting with Essenia fetida Rumbricus ribeus. The duration of this vermicomposting was 2 months. Some pictures from the process and the composting process here, and this is the final product, but this is the vermicomposting. So we found, studying the prokaryote and eukaryote, we found quite different diversity in each stage. We observed the source materials. After that, the first mesophilic phase, thermophilic phase, second mesophilic phase, and the microbiome and the prokaryote in the compost. As you can see, the diversity is quite different. The structure of the prokaryote is quite different, but we can say that there is a quite similar species between the final compost and the second mesophilic phase. At genus level, the situation is similar, quite significant similarity in the during the maturation in second mesophilic phase and the final composts. So the alpha diversity is shown here. The highest Shannon index we observed in the first mesophilic phase, but the highest number of species were found in the final compost. On the left side is the Venn diagram, which is quite useful to compare the taxa in this case, genus level in this case, and the overlapping of the genera in this in different samples in different phases. So we can say that we have overlapped 916 genuses, but also there are different similarities mostly between the compost and the second mesophilic phase, that are 388 genera. So, and here is the cluster tree based on the weighted unifrac distance. So that confirm our findings till now. Speaking about the eukaryote microbiome, we can say that the situation is a little bit different. We have a lot of Ascomycota at phylum level in all stages, but here in first mesophilic stage it's different because we have a lot of Moderator also we have different phylums. This is confirmed by the findings in genus level, but from the alpha diversity, we can say that again we have increased alpha diversity in the compost, final compost, but also in the thermophilic and mesophilic stage. Here we can observe the species inside of each in each phase of composting, and considerable decrease of number of species in the second mesophilic phase. So again, the Venn diagram and the cluster tree of the eukaryote. Here we have much less number of taxa at genus level. So we can make a short conclusions that the temperature had a major role in the composting bacteriome changes, influencing positively species abundance, Shannon and Simpson indices, and negatively ACE and Chow indices. Firmicutes and actinobacteria dominated the prokaryote in the source materials and in the thermophilic phase, while Proteobacteria was the dominated phylum in the both mesophilic phases and in the final vermicompost. Moderator were the most presented fungi in mesophilic phase, while the thermophilic Ascomycota and Basidiomycota were dominating in this stage. We produce one paper in this from this experiment, and preparing another one. Now, shortly, because the time is flying, as to say, I would like to present our work on the field experiments applying different organic and mineral fertilizers in two fields. This is the one of the experimental fields of Agricultural University of Plovdiv. We had two fields, one with traditional crop rotation and the other one with some new aspects of crop rotation. these new aspects are, for example, in traditional, we start with growing oats, while on the other field, we grew oats with veg together. And after that, in the flowering, we plowed the oats and veg mixture as green manure into the soil. In the same time, we used during the second year on the maize a second crop. In this state, we use maize and soybean together as example of intercropping. So in each field, we applied 5 treatments: control, untreated, mineral fertilizer as NPK, vermicompost. We produced earlier mineral fertilizer and vermicompost and biochar supplied from our colleagues in ConnectFarms project from Poland. The University of Life Science is also our partner in UNIgreen. This is the soil characteristics, that is slightly alkaline pH, and here are the vermicompost analysis and biochar analysis. So biochar, we have 65% of total organic carbon. Some word about intercropping. So according to the literature, the intercropping is conducts to different benefits, yield enhancement, greater resource use, such as increase of the soil nutrients, soil moisture retention, etc. Improving the soil biodiversity, and applying legumes and another crop that supports the main crop growth. So the root system of intercrops, legume and cereals with different exposure areas, allowing experimentally complementarity to avoid root-to-root competitiveness. And of course, the intercropping is very important because the growth promoting rhizobacteria colonize the rhizosphere and many plant species and confer beneficial effects on them through direct or indirect way. So maybe you know the plant is very important source of nutrients because it loses about 40% of photosynthetic carbon through its roots. So combining different plants in the same place, you combine also their microbiome, so microbiome and also endophytic microbiome. So the aim of our study was to determine the influence of two factors on maize yield and on soil microbiome activity and diversity: the crop system from one hand, and from the other hand, the kind of soil amendment. We have two cropping system: single cropping, as to say the maize alone and the soybean alone, and intercropping both together in a row. Row intercropping of maize and soybean as second crop after barley. You can see here the row where maize and soybean grow together. The sowing was during the mid of July 23. and harvesting in mid of October. The sowing was done after barley. Another pictures of our experiments. Here they are combined. Here they are grown one culture alone. So the inoculation rates of the soybean seeds with Rhizobium japonicum, and the aspects of uninoculated and inoculated seeds. So some agricultural findings that really we think are important. As you can see, this is the yield of maize in the end of during the harvest. You can see here without applying fertilizers, we can obtain a significant increase of yield of maize here in the control. Highest results were obtained in case of intercropping, applying mineral fertilizer or combined fertilization, while in the vermicompost case we don't see this, this effect. The positive effect of intercropping was also seen studying the 1000 grain mass in case of mainly in case of mineral fertilization. The pH almost in all cases where intercropping was applied in case of maize were decreased the pH. Small decrease, but it was significant. And concerning the electrical conductivity, we found this phenomenon in case of vermicompost, applying vermicompost in case of maize. And here also in this biochar and combined treatment. Concerning concerning the microbial activity in soil expressed as basal respiration, we can say that vermicompost in shown different shown the same increase in different treatments, but it was depending on the application of vermicompost, not by the cropping systems. The situation was similar concerning the substrate induced respiration, which is an indicator of potentially active soil microorganisms in this case. The case of glucosidase is similar. Vermicompost shown the increased glucosidase activity, but it was very interesting that in the control, in case of maize rhizosphere, the glucosidase activity was also increased. So here we can say some words about the prokaryote of these two cropping systems and applying fertilizers. So on the first place, here you can observe the bacterium of soybean and maize is similar in case of applying compost, but in the traditional growing of maize, it was different because you can see that the same genera are not dominated here. The dominated bacterium is quite different. The situation is the same in case of biochar. Here we have the bacterium of soybean and maize, and here of the traditionally grown maize, and also the maize in case of mineral fertilizer application. And here we have the traditionally grown maize with mineral fertilization. We can observe here some overlapping of species. Here we have principal component analysis at genus level and species level, and depending on the kind of agricultural system, we have different clustering. For example, the traditionally grown crops and their microbiome are here. Intercropping is grouped together, and they are differ depending on the cropping system, as you can see here. And the situation is similar at genus level, at species level, sorry. Here are the clustering tree based on Bray-Curtis distance in species abundance. The clustering is quite similar, and I only would like to highlight here in this grouping that is traditional maize and here is the traditional growing and here is the intercropping. That is, they are grouped together differently. You can see the traditional with applying mineral fertilizer is somehow different from the other group of bacterium. This is the principal coordinates analysis based on the abundance of species level. Also, we can observe the grouping, the clustering. It's of intercropping of the maize, and here also the maize traditional, the bacterium of maize in traditional growing. The Shannon index were found increased in case of maize growing in intercropping system, applying mineral fertilizer, and also applying biochar. Species abundance, on the other hand, were increased in case of soybean in intercropping case, and in both cases of intercropping when applying biochar. We can say that we found highest in this heat map, highest alpha diversity in case of biochar in intercropping, applying biochar in intercropping, especially in the maize rhizosphere. The incorporation of vermicompost in case of intercropping is closely related with the main microbiome activity expressed as basal respiration, starch-induced respiration, and glucose activity. So the main conclusion is that intercropping led to enrichment of the rhizosphere microbiome and improvements in physico-chemical parameters, resulting in high maize yields. We can say that we published for that 3 or 4 year periods almost 11 papers. Most of them are in repeated papers, repeated journals like agriculture, microorganisms. So this was from my side. Thank you for your attention. And last but not the least, I would like to thank all the participants in our team from our university. We are from 5 different departments. Thank you all of you colleagues for your hard work, and you can check some of our results and papers in these sites. One is in our university, the other is the official site of the project. Thanks a lot.
Thank you very much, Stefan. That was very insightful presentation. Do we have any question from our audience? You can ask the question in conversation or in Q&R, or if you are unmuted, you can speak now if you want. Or maybe I can start meanwhile. Stefan, I was wondering how much from the microbiome comes from the gut microbiome of the worms in the vermicomposts.
This is a good question. We can say that it is not studied by us till the morning, till now. But we can say that, of course, we have to publish another results that are not still prepared for publication. And this is a very good appointment because we know that the rooms, they are very important as source of microbes. And it could be very useful to look how the soil microbiome was changed applying this vermicompost, because there are different, quite different microbiomes applying vermicompost. It is completely changed after applying this biofertilizer. Yeah.
Mm-hmm.
Thank you for this question, Patrick. It was, yeah, it's really interesting.
Yeah, that's interesting. And it was I had the second question, but you started answering it about the impact on the original soil microbiome. Because in your introduction, you mentioned that with our agricultural practice, we are modifying microbial biodiversity, and in particular, pesticides are affecting microbial biodiversity. But I was wondering to which extent compost alters the soil microbiome.
Speaking with numbers, I think on memory it's about 30% difference when applying vermicompost with where we don't have any additives, any amendments in the control, because the situation is with the other treatments comparing with the control, because we added in the other treatment, we added mineral fertilizer, but when we add combined fertilizer, I mean mineral and organic, I mean vermicompost, the situation is improved, and it is you can see it from the results. That's the difference in there is not too big difference in the Venn diagram when you I'm very sorry it is not presented here, but the Venn diagram in this case shows that there is a similarity between both treatments, vermicompost and combined treatment.
Mm-hmm.
And this is really, as you said, this is the importance of these cropping systems on one hand, and on the other hand, application of organic matter and recycled organic waste.
Mm-hmm. Yeah. I was also surprised by the result that you showed, where when you use vermicompost, there was no increase in yield, maize yield, in the second study.
Yeah.
How do you explain this compared to other treatment? Other treatment were increasing the yield, and in the case of vermicompost, there was no increase of yield.
Yes,
If I get it right.
Yes, I think I will just open the presentation. Maybe you mean these results?
Yes, right. Yeah.
Yeah, no, we have some increase, especially in the traditional, not in intercropping case. We can say that we have here more than 20, even 30% increase from this to this, applying vermicompost, but the results, they are not quite higher to say that the results are the best in this case.
Mm-hmm. And we see that when we compare intercropping and traditional for vermicompost, we do not see the increase that you
Yes, we
here.
think that this is due because the application of vermicompost by one hand and the combining and the intercropping, because I think the microorganisms and the plants already have nutrients, but the problem is I think from the point of view of dominated microbiome of vermicompost, because maybe this is in this case we can say that maybe there are some competition in in the soil microbiome, so that's why we don't see
Mm-hmm.
some this increase as in case of mineral fertilization and combined fertilization in this case.
Mm-hmm.
Maybe in the other side, when we repeat this experiment, the results will be more clearer because we applied here a crop rotation system, so with 3 years duration. So in this case, the application we will follow.
Mm-hmm. Yeah, this hypothesis about competition is interesting, and maybe it's reflected also in the measurement for respiration that you made. You show that there is an increase of respiration, which means that there is a increased activity of microbial life, which might be consuming the nutrient and so on, and therefore there is competition with the plants.
Yes, this is one of the one possible reason, but yes, the truth is that we have to apply more efforts in studying this phenomenon, because Or really the nutrients there, the application was, I think, let's see, we applied, I think, not a big concentration of vermicompost. So I'm not sure why these results are appeared, because the quantity of compost is not were not too high. I think it was 2 tons per hectare, or no, I mean 20 tons. I'm really not sure at that moment. In the beginning, I show in the beginning. Let's see. Here we have 12 tons per 12 tons per hectare. Yeah. While the biochar were 10 tons per hectare. So it's not big fertilization. Yeah. Normally we can apply to 20-30 tons per hectare without any problems. Yeah.
Okay. Don't hesitate to ask questions in the audience. Otherwise, I can go on. So overall, Stefan, do you think that the fertilization effect comes from the microbiology, so the additional microbes that you are adding with the compost, or from the nutrients that have been processed in the compost, or it's a mix of both? Is there a way to quantify the how
I think
these
it's
parameters
multiplying effects. It is multiplying effect, and actually this is the crop grown experiments that we still didn't have published, although we have published another experiments. I think it's maybe it will be clear after preparing the papers for publication, but really I think it's multiplying effects of both crop rotation, because we have here green manuring and cropping system, intercropping the maize and soybean, and additionally the fertilizer. So in that sense, because we have by one hand combination, the maize has additional nutrients from cooperation with soybean, and from the green manure in the beginning, in the previous year. So that's why we think that the rule of applied fertilizers are not very evident. But in control, we can see some difference, as I showed here. And we think that this is very important in that case, because without fertilization, we have increased with 19% of yield of maize here, instead of traditional growing of maize alone. And this is the control without applying fertilizer. Also, we have to take in mind that this is the growing maize as a second crop. So after harvesting barley, so maybe this also influence the results.
Okay, yeah, that's already a lot, a big difference. There is a question, Stefan. Do you know the composition of the compost? So I guess it's both mineral and biological composition.
Yes, I think it was presented in the beginning. So here we have physical-chemical composition. So pH 7.8, total organic carbon 18, and nitrogen we have 1.27%. The soil, I forgot to say, that was very poor of nutrients, especially phosphorus and nitrogen. Speaking about the compost composition, we have here the composition. It's this is its order level, I think, and here is a genus level, the compost composition. So we have mainly Sodariomycetes here, the blue one, Moderator here, and another also Agaricomycetes here also. Speaking about the proteome, at genus level, they are mostly Ralstonia, and Bacillus. But of course, there are many more, but these were dominated species, genera that were found in case of prokaryote.
Okay. Another question would be is, do you look at the correlation between compost composition and yield? So you show that this has been done with one type of compost or many different type of compost?
We've done in one type of compost and one type of biochar, but biochar was studied as we added only once the biochar, and we studied the effect of biochar during 2 years. It was added before the barley growing and sowing, and we didn't add any additional nutrients till the end of crop rotation. So concerning the vermicompost, we used only this one. Yeah.
Okay. May I ask maybe one last question, because you showed here the taxonomy composition of microbiome. I was wondering whether you were interested by the functionality. Did you look at the enrichment of genes in this microbiome? Maybe there are some genes that are more enriched for nitrogen mineralization and so on.
We have the results because this study was done with shotgun analysis. So,
okay. So you have the data.
We have the data, but I decided not to present because there are a lot of data,
Yeah,
and
sure.
the analysis is very important. So I would like to thanks again to our team from the University, because of they did a lot of job here. Yeah. About the field experiments, laboratory analysis, etc., etc.
Okay, so with this, I don't see any more questions. I would like to thank you again, Stefan, for this very appealing presentation. That's the way we have to go in the future, for sure, to be more relying on natural ecosystemic services delivered by microbes, if possible. And hopefully we will be successful in this.
Yes, thank you, Patrick. We are working on other project also, so we are open for collaboration with your university and the UNIgreen or with other colleagues to strengthen the collaboration and to involve more students in our research. Thanks again. Thank you for all of you. Thanks.
Thank you very much. So to finish, I would like to share a QR code just for a survey. And if our participant would take a bit of time to complete the survey, we would be really grateful for this. Okay. So again, thank you very much, Stefan. Thank you to the audience, and I hope you will be interested by the next webinar to come. Thank you.
You.
Bye-bye.